Today I am going to discuss the purine nucleotide cycle, which is very important metabolic pathway that supplies energy for the exercising muscle during high intensity exercise such as sprinting or weightlifting. Sprinting is basically running very fast for 100, 200 or 400 meters. So I will like to give you first brief background. When we are doing high intensity exercise, body needs energy in the form of ATP. This cycle or purine nucleotide cycle is an important metabolic pathway that supplies energy utilizing purine, particularly AMP or adenosine monophosphate, to produce energy. That is why it is also called as AMP cycle. In this video, we will explore the mechanism, regulation, and significance of this cycle during high intensity exercise. Therefore, I welcome you to this video on purine nucleotide cycle. Please watch it till the end. What is purine nucleotide cycle? This is also known as AMP cycle because AMP is the substrate and the product of this cycle. It was described first in 1972 by James Lowenstein in skeletal muscle. It completes in the fast twitch muscle cell cytoplasm in 3 steps, utilizing 3 key enzymes. This is an energy dependent process utilizing GTP as the source of energy. It is activated by high AMP concentration to produce ATP for muscle activity, and it also produces ammonia, which buffers acid which is produced during exercise in the muscle. Excess of ammonia is also removed by the liver using urea cycle. Let us examine energy pathways available in skeletal muscle during intense exercise. During intense exercise, skeletal muscle relies on rapid energy sources. First, stored ATP provides energy for 2 seconds. Next, phosphocreatine generates ATP for up to 10 seconds. As exercise continues, anaerobic glycolysis becomes the primary energy source for short-term use, typically lasting up to 2 to 3 minutes, after which aerobic metabolism predominates. Anaerobic glycolysis also produces lactate. Additionally, as ATP is depleted rapidly and the AMP level is increased, the purine nucleotide cycle is activated. This cycle helps maintain energy balance and produces fumarate, which enters the TCA cycle to support prolonged muscle activity. Let us examine the mechanism of purine nucleotide cycle. In the skeletal muscle, during intense exercise, ATP is consumed, producing ADP. ADP is converted into ATP and AMP by the action of adenylate kinase enzyme. This enzyme is also known as myokinase and is highly active in skeletal muscle. This reaction is reversible reaction. However, the product of this reaction, AMP, is consumed in the purine nucleotide cycle, thereby pushing this reaction in the forward direction, making ATP available for the muscle activity. AMP is converted into IMP or inosine monophosphate and ammonia by the action of enzyme called AMP deaminase. This is the first step of this cycle. Ammonia basically buffers the system and excess of it is removed as urea in the liver, produced in the liver. Therefore, you expect to see high level of urea in the urine of exercising individuals. In the second step, IMP condenses with aspartate and is converted into adenyl succinate by the action of enzyme called adenyl succinate synthetase. This enzyme utilizes GTP as the source of energy. In the third step, adenyl succinate is converted back into AMP and fumarate by the action of adenylate succinate lyase enzyme. AMP produced during this reaction supports the continuation of the cycle, thereby pushing the adenylate kinase reaction in the forward direction, making ATP available for the muscle activity. In addition, fumarate joins the TCA cycle and support the production of energy for muscle activity. Therefore, purine nucleotide cycle maintains energy balance during intense exercise by removing AMP and generating fumarate, which enters the TCA cycle and supports ATP production via electron transport chain and gluconeogenesis. I have already presented a video on gluconeogenesis which you can watch at your convenient time. We have seen regeneration of AMP during purine nucleotide cycle. This regeneration of AMP causes its accumulation. This inhibits the enzyme adenylosuccinate synthetase by allosteric mechanism. This block causes increase in IMP level or inosine monophosphate level, which is finally degraded into uric acid. This degradation is critical to restore AMP level and ATP production by AMP kinase. Thus, in intense exercise, hyperuric acidemia is transiently present. The cycle is significantly important because it supplies ATP for the exercising muscle. In addition, it also produces fumarate, which further supports energy production through electron transport chain and also through gluconeogenesis. In addition, it produces ammonia, which buffers acid, which is produced during exercise in the muscle. Impairment of the cycle is associated with exercise intolerance and muscle fatigue. These symptoms are also commonly present in AMP deaminase deficiency, which is a genetic condition. In adenylosuccinate synthetase deficiency, it is very, very rare, and this is also associated with the impairment of the purine nucleotide cycle. In adenylosuccinate Ia deficiency, there are neurological symptoms such as seizures and autism. Thank you for watching this video. Please like it, share it, and subscribe it. Until next time, you take care. Bye bye.